The topic of today's presentation is vitamin A. The term vitamin A or preformed vitamin A is refers to retinol, retinal and retinoic acid. Retinol alcohol form, retinal aldehyde form, retinoic acid not active in vision. Retinol is present in food as fatty acid esters. The most common among them is retinyl palmitate. Retinyl esters are formed in animal tissue, primarily egg yolks, butter, whole milk products, liver, fish liver oil. The term provitamin A refers to beta carotene and other carotenoids which can be converted to retinol. Compounds providing vitamin A actively are called carotenoids. These are also called vitamin A precursors, found in plant only. Carotenoids, the orange and yellow pigment, are synthesized by plant in wide variety. The pigment with the most vitamin A actively in beta carotene. Two other commonly occurring carotenoids are alpha and gamma carotenes. Conversion of carotene to retinol. The conversion of beta carotene into retinol occurs inside the intestinal mucosa and to a very small extent in the liver. Retinal is first formed with the cleavage of beta carotene. Retinal is then converted to retinol. Some retinal is oxidized irreversible to retinoic acid. Beta carotene can produce two molecules of retinol, but in vice versa, this does not occur. 6 microgram of beta carotene equal to 1 microgram retinol, that is equal to 1 Re or 1 retinol equivalent. Up to 30% of beta carotene may leave the intestine without being converted to retinol because the activity of the conversion enzyme is low. Digestion and absorption. In a mixed diet, protein bound retinols, ester, and carotenoids are hydrolyzed by enzymes in the intestinal lumen. Retinyl esters are then converted to free retinol by esterase and lipase. Carotenoids and free retinol, along with dietary lipids and bile, are incorporated into micelles and get inside the enterocytes. The efficiency of absorption differs between preformed vitamin A and carotenoids. Approximately 70-90% of dietary preformed vitamin A is absorbed. Dietary carotenoids absorption varies widely, 5% from uncooked vegetables to about 60% if present as pure oil or as a part of aqueous dispersion in supplements. Initially, retinol is esterified in the mucosal cell to form retinyl palmitate. These newly formed esters, along with N carotenoids that are absorbed unchanged, are incorporated into chylomicrons and carried via lymph into general circulation. Chylomicrons remains deliver retinyl esters and a part of the carotenoids to the liver. The other portion of the carotenoids is deposited in the fat cells along with fatty acids removed from chylomicrons for storage. Absorption of vitamin A. Storage of vitamin A. Retinyl esters reaching the liver is hydrolyzed to retinol. Retinol can combine with cellular Retinol can combine with cellular retinol binding protein CRPB for transport to tissue. It can be esterified and stored. Vitamin A is stored in special liver cell called stellate cells along with other lipids droplets. Only after the stellate cells can accept no more retinol for storage does hypervitaminosis A occur. Thus, plasma vitamin A level remains constant over a wide variety of dietary intake.